हो का, हे आले तू.
सर पीपीटी वापरून सेट करेल ना.
काहीच नाही, 4:30.
कोणी आज तिकडे सर तू दुसरं आहे का ते व्हॅलिड
सीईटी चालक कोणाकडे?
गौतम सोबत कोण आहे त्याच्याकडे.
पाटवेकर सर बुटले सरांचा आपण मेल घेतला, पेपर घेतले, पण त्यांना रजिस्ट्रेशन मध्ये नाही टाकले.
त्यांनी हेच केलं नाही,
रजिस्ट्रेशनच केलं ना,
पैसेच भरले नाहीत त्यांनी.
हो, म्हणून स्कूल ऑफ लाईफस्टाईलची वेगळीच लिस्ट टाकली त्यांना शेवटी त्यामध्ये.
फोल्डर
हा फोल्डर
आणि पार्टिसिपंट ऍड करत जायचं. युअर
तिथे लावशील का बेटा असं तिथे टेप लावलेले ते या बाहेर पण धावत पळत का आला
चल लवकर 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 चालत. पटकन पटकन लोक काय करतात, काय वाजलं. हे लोक जे येतात ना ते ऍब्सोल्यूट इथपर्यंत आणा आहे त्याच्यापर्यंत. मेक इट फर्म.
It was originally known as Shatatantri Veena, which means 100-stringed lute. The name Santoor was given by the Persians. Similar instruments are found all over the world, like Santoor in Iran, Yangqin in China, Cymbalum in Hungary and Romania, Santuri in Greece, Hackbrett in Germany, hammered dulcimer in certain European countries and America. Santoor is played with a pair of curved sticks.
Hello? 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 Welcome, sir. Welcome. Good morning to all dignitaries, guests, and delegates with a great joy and immense exultation. A very warm welcome to one and all present over here. I, Dr. Amrita Kanakarande, feel privileged to welcome you all on behalf of Organizing Committee of 3 days International Conference on the theme of Advances in Bioactive Molecules, ABM 2022. Well, Albert Einstein said, if we knew what it was we are doing, it would not be called as a research, would it? It is on the occasion like this and understandings. We live to get an exposure about what's the base of the brains think about bioactive dynamic biomolecules used for versatile applications. Novel bioactive compounds obtained from nature's biodiversity and targets in fields like biology, environment, food, public health, and medicine, etc. With a dimension spanning from nanometers to micrometers and used for versatile applications. With this platform of ABM 2022, which encourages crosstalk between diverse domains to find sustainable solutions in healthcare and allied fields. Now, I take this opportunity to introduce our today's guest of the inaugural ceremony. We have with us Honorable Vice Chancellor of SRTM University, Dr. Uddhav Bhosle, sir. I also welcome Pro Vice Chancellor of our university, Jogendra Singh Bisen, sir. I also welcome Dr. L.M. Wagmare, sir, Dean, Faculty of Science and Technology of SRTM University. And I also welcome Professor R.S. Mane, sir, Director, IIL. I am profoundly delighted to take an opportunity to welcome as a chief guest for this inaugural session. We have with us Dr. Baban Ingole, sir, visiting National Center for Polar and Ocean Research, Goa. We have with us Dr. R.V. Omkumar, Neurobiology Division, Rajiv Gandhi Center for Trivandrum, Kerala. They are the aspiring figures of all of us. We feel fortunate enough to have them among us. I also welcome Professor H.R. Sarma, sir, Department of Zoology, Rajiv Gandhi University, Arunachal Pradesh. Also, I welcome all the eminent scientists who have joined virtually for this conference. And also, it's my pleasure to extend my cheerful welcome to our Director, sir, School of Life Sciences, Professor S.P. Chavan, sir. Welcome to Dr. Pathak, ma'am, convener of this international conference. I also welcome co-convener of this international conference, Professor T.A. Karam, sir, and Dr. B.S. Survase, sir. I also welcome Dr. Arya Mulani, sir. It's my pleasure to welcome all eminent speakers who joined different corners of the India as well as abroad virtually for this conference. I also welcome all the organizer committee members, advisory board members, technical committee members, and publication committee members. And I also welcome different school directors and respected faculty members. One and all, very warm welcome. And last but not least, it's gratifying welcome to look around us and see so many familiar faces of research scholars, dear students, after almost 2.3 years of this pandemic situation. At last and not least, I would like to say thanks for esteemed audience we have gathered over here and joined virtually from various places, from loops and corners of every part of the globe. As light symbolizes victory over darkness and defeat ignorance, it marks the beginning of auspicious events and moments. I request all dignitaries on the dais to garland the photo of Swamiji and inaugurate the 3 days international conference on advances in bioactive molecules, ABM 2022 by lighting the traditional lamp. Please, sir. We'll start Saraswati Savan on this auspicious occasion. Jaya Sharade Vageshwari, Jaya Sharade Vageshwari, Vidhi Kanyake Vidyadhari, Vageshwari, Vageshwari, Jaya Sharade Vageshwari, ज्योत्स्ने परी कांती तुझी, ज्योत्स्ने परी कांती तुझी, मुखरम्य शारद चंद्रमा उजळे तुझा, हास्यातुनी उजळे तुझा, हास्यातुनी चारी युगांची पौर्णिमा, तुझ्या कृपेचे चांदणे, तुझ्या कृपेचे चांदणे, नितवर्षुदे अमुचा शिरी, नितवर्षुदे अमुचा शिरी, वागेश्वरी जय शारदे वागेश्वरी, वीणेवरी फिरता तुझी, वीणेवरी फिरता तुझी, चतुरा कलामयंगुली, संगीत जन्माये नवे, संगीत जन्माये नवे, जडता मतीची भंगली, उन्मेष कल्पतरुवरी, उन्मेष कल्पतरुवरी, बहरून आल्या मंजिरी, वागेश्वरी जय शारदे वागेश्वरी, जय शारदे वागेश्वरी. Thank you one and all. Thank you. Thanks to all dignitaries once again. To welcome is to show honor. To welcome is to establish integrity. Now, may I request Professor S.P. Chavan sir, Director, School of Life Sciences, to welcome our Honorable Vice Chancellor of University, Dr. Uddhav Bhosle sir, by offering a token of love. Thank you, sir. Thank you, sir. I would like to request Professor T.A. Kadam, sir, co-convener of this conference, to welcome Pro Vice Chancellor of our university, Dr. Jogendra Singh Besen, sir, by offering a bouquet. Thank you, sir. Thank you, sir. I would like to request Dr. L.S. Kamble, sir, to welcome Dr. L.M. Wagmare, sir, Dean, Faculty of Science and Technology of our university. Thank you, sir. Thank you, sir. Now, I would like to request Dr. A.P. Pathak ma'am, convener of this conference, to do the honor of our today's chief guest, Dr. Baban Ingole, sir, visiting scientist, National Center for Polar and Ocean Research, Goa, by offering a token of love. Thank you, sir. Thank you, ma'am. Now, may I request Dr. G.B. Zore, sir, to do the honor of today's chief guest, Dr. R.V. Om Kumar, Scientist, Neurobiology Division, Rajiv Gandhi Center for Biotechnology, Trivandrum, Kerala, by offering a token of love. Thank you, sir. Thank you, sir. Now, may I request Kodgire ma'am to do the honor of chief guest, Dr. H.N. Sarma, sir. Professor, Rajiv Gandhi University, Arunachal Pradesh. Thank you, sir. Thank you, ma'am. Thank you once again, all faculty members, respected dignitaries on the dais. Thank you very much. Now, may I request Professor S.P. Chavan, sir, Director, School of Life Sciences, to introduce our school and give opening remarks of this inaugural ceremony of this international conference. Sir, please.
The dignitaries on the dais, Honorable Vice Chancellor, Sir, Uddhavji Bhosle, our Pro Vice Chancellor, Sir, Yogendra Singh Bisen, Sir, the Dean of Faculty of Science, Dr. L.M. Wagmare, Sir, the guest invited for this conference, Professor Om Kumar, and other guests, Dr. Baban Ingole from NCPOR Goa, then Dr. H.N. Sharma, sir, from Rajiv Gandhi Central University, Itanagar, Arunachal Pradesh, the all faculty members of this university, dear students, and the media person, and all dear colleagues. So it's a great pleasure for School of Life Sciences to host this international conference, which will be held during 7th to 9th of this month. And it is with the full of technical sessions. And we called here, it's a great feast for all students to listen. Very well-known, world-class resource person and the experts in biomolecules. So this is second event, international event, which is being hosted by this School of Life Sciences of this university. So the theme is very particular, very precise, and that's why the experts in biomolecules only they send the abstracts and the papers. Nearly 100 abstracts we received. Similarly, full-length 50 papers we received. These will be published in the UGC Care Listed Journal, as well as remaining papers, selected papers will be published in a reviewed book as book article. So the response is very good, but as the situation of the COVID has not fully, as we expect, has not fully covered, and therefore around half of the participants are online, and the presentations are also online. So with this opening remark, I welcome all again for this event. They will enjoy the technical sessions. Major sessions are here. Online sessions will be at School of Life Sciences. So with this, I conclude my opening remark. Thank you very much.
Thank you very much, sir. Thank you, sir. Now, may I request Dr. A.P. Pathak, ma'am, to say a few words on this occasion, please, ma'am.
Good morning, one and all. Honorable Vice Chancellor, sir, Honorable PVC, sir, and all dignitaries on the dais. Now I request our keynote speaker, Om Kumar, sir, please declare that this function has been inaugurated. Om Kumar, sir.
Hello.
I'm really honored to do this job. I declare that the conference is inaugurated. All the best wishes for the conference and for very active scientific delegations. Thank you.
Now I request Honorable Vice Chancellor, sir, sir, please inaugurate that and publish the abstract.
Thank you very much, sir. Thank you, all dignitaries on the dais. Thank you very much, sir. Now, may I request Honorable Vice Chancellor of our university, Dr. Uddhav Bhosle, sir, for presidential address and declaration of inaugural ceremony. Sir, please.
Very good morning to one and all. The dignitaries on this dais, Professor Bisen Ji, Pro Vice Chancellor of this university, Dr. R.V. Omkar, Dr. H.N. Sharma, Dr. Baban Ingole, my colleagues at this university, Dr. L.M. Wagmare, Dean, Faculty of Science and Technology of this university, Professor S.P. Chavan, Professor Pathak, of the dais, my teaching colleagues from different schools on campus, non-teaching colleagues, student, media person, ladies and gentlemen. Once again, very good morning to one and all. It is my privilege to welcome dignitaries on the dais and off the dais for this inaugural function of International Conference on Advances in Bioactive Molecules. It is also my privilege to be amongst you, among this gathering this morning. I extend my congratulations to Professor S.P. Chavan and his team for organizing International Conference on Advances in Bioactive Molecules. At the same time, I am also thankful to different institutions, resource persons for accepting our invitation and for their active participation in organizing this international conference. This university, established in 1994, has achieved a remarkable progress in the field of academics, research, and extension activity. It has name and fame at state, national, and international level for its contributions in the various fields. This university has around 275 plus affiliated colleges at Latur, Nanded, Pervani, and Hingoli district, offering around 167 programs at undergraduate and the postgraduate levels. We have a student strength of 1.75 lakhs, and efforts are on to increase the enrollment of the student in the higher education. To increase the enrollment of the student in the higher education, as a part of it, this university has established a model degree college at Hingoli in a district where GR is very low compared to the GR of other states or other districts in the states. This university has 13 schools on campus and three schools at sub-campus Latur and 3 schools at sub-campus Pervani and constituent degree college MDC Hingoli and the research centers at Kinvert as well as research centers on the campus. University has, as of now, 67 international students from 5 different countries, and efforts are on to increase this number and the nationality in near future. This university is known for an research and extension activities in various fields. University has created a state-of-art laboratories through the funds that we have received from different funding agencies, at the same time from the funds from the university own funds. Efforts are on to ensure that the state-of-art facilities will be created on the campus, which will be made available for the student and the researchers of not only campus, but the affiliated colleges too. In line with the same, university intend to develop a common central instrumentation facility where the basic instrumentations that is required in physics, chemistry, mathematics, biology will be housed under one roof, which would be a state, which would be the facilities that will definitely enhance the research culture here. At the same time, university intend to develop a common computing facility where the computing facility where the competition can be possible one. This university, in order to promote the research and academics, or I would further say that in order to promote interdisciplinary research, this university has established 2 research centers in other state. And I am very proud to say that this is only the university in the state of Maharashtra which has research centers in other states. So university has a research centers at INCOIS Hyderabad and other one at AMD, Atomic Mineral Divisions, Department of Atomic Energy, Government of India. So these 2 research centers will definitely enhance the research culture of this university. I am very confident. At this point, I would also appeal all the students and the teachers of this university to take the maximum advantage of the facilities that have been that are possible due to the research center establishment of this university. This university offers choice-based credit system and have established or instituted a policy for the credit transfer. The student of this university can opt any course which is offered at other universities at the same time which is offered at platforms such as NPTEL as well as the SWAYAM platform. Sir, I would like to say that in the last 2 years, 25% student from the campus opt courses that are offered either on SWAM platform or NPTEL platform. University has made a special provision. University is extending a special assistance for affiliated colleges to establish local chapters to offer the MOOCs courses that are offered on the different platforms. It means that our university is ahead, which always foresee what would be the future. The choice-based credit system, at the same time, the availability of the MOOCs courses, which is the basic requirement or which is basic in national education policy. But this university itself has started all these activities, I would say that 3 years before the commencement of or implementation of a national education policy. In all the aspect, this university is far ahead in many sense. The researchers as well as the teachers of this university have a great recognitions, and many of them have been awarded at national and international level for their contribution in the field of research and extension. I hope that this culture of research will further strengthen at this university, and from administration side, a lot of efforts are going on to ensure that the research culture will again strengthen, which will benefit not only the student and teachers on the campus, but affiliated colleges too. In future, our efforts are on to ensure the multidisciplinary research culture on the institutions, as well as on the campus, as well as in affiliated colleges. This international conference on advances in bioactive molecules will definitely prove to be useful, not only for the researchers in the field of life sciences, but equally important for the researchers in the field of chemical sciences, pharmaceutical sciences, as well as other sciences too. I would further say that this international conference will definitely provide a platform, has provided a platform for interdisciplinary research, and which is the need of the today, I would say. So with this, before I conclude, once again, I congratulate Professor Chavhana and his team for organizing this conference, and I hope this conference would be successful. Successful in the sense, the aim of the conference will definitely achieved, or outcome of the conference will be definitely achieved, which will be helpful for the growth of this school in a longer run. Thank you. Thank you very much.
Thank you, sir. Thank you very much, sir. Your thought-provoking address set a perfect platform for our speakers to deliver their presentations in the new era of bioactive molecules for versatile applications. Encouragement, enthusiasm, and excellency is one of the major aspects of the organization, and we are continuously receiving these things from our Honorable Vice Chancellor, sirs. Thank you very much, sir. Thank you. Now, I request Dr. Hemalata Bhosle, ma'am, to propose vote of thanks on this inaugural ceremony. Ma'am, please.
Good morning, one and all. Thank you is such a prayer that can't be seen or touched. It can be my heart. I feel privileged to propose vote of thanks on this occasion of 3 days international conference on advances in bioactive molecules 2022 on behalf of entire organizing team. I am deeply grateful to the Honorable Vice Chancellor of our university, Dr. Uddhavji Bhosle sir, for presiding over this conference or this event and for enlightening address. I am also thankful to the Pro Vice Chancellor, Dr. Jogendra sir, Registrar Dr. Sergey Rao Shinde sir, Dr. Yalam Wagmare sir, Dean, Faculty of Science and Technology, this university, Finance and Account Officer, Dr. Anand Barpute sir, for helping us in organizing this event. I take this opportunity to express our sincere thanks to the respected Dr. Om Kumar sir for gracing this occasion. His presence today from such a long distance from Trivandrum, sir has came here. So his presence today in this function has immensely enhanced its importance. We are extremely grateful to you, sir, for accepting our invitation. I must express my deep gratitude to our eminent guest, Dr. Babaningole sir, Dr. Yachan Sharma sir, and Dr. Yogesh Shauche sir for giving us precious time in spite of busy schedule. It really means a lot to us. I'm also thankful to all distinguished members in the campus and of the campus for supporting us to organizing this event, especially Professor R.S. Mane sir. Since beginning for organizing this event, sir has supported a lot to us. So thank you, sir. Last not but the least, I extend my sincere thanks to all the delegates and participants who have made this convenient to attend this conference in presence or in virtual mode. I also thankful to media personals and press personals for having time to cover this event. I also wish to thank all the members directly or indirectly who are involved in organizing this event. I just want to say that have a wonderful staying at this campus and be with us for next 3 days to make this event more successful. Thank you.
Thank you, ma'am. Thank you, everyone. Now, may I request Dr. G.B. Zore, sir, to please introduce our today's chief guest, Dr. R.V. Om Kumar, Scientist, Neurobiology Division, Rajiv Gandhi Center for Biotechnology, Trivandrum. Sir, please.
Very good morning. So this is my privilege and I feel honored to introduce Dr. R.V. Om Kumar sir, presently serving as a scientist G at the Rajiv Gandhi Center for Biotechnology, Tirunananthapuram. He is heading the Department of Molecular Neurobiology Division of Rajiv Gandhi Center for Biotechnology. Dr. Om Kumar sir, MSc in biochemistry, and he completed MSc from Kerala University in 1986. He has completed his PhD from Indian Institute of Science, Bangalore in 1992. And we are really honored to have a person of such a caliber with us. And Dr. Om Kumar sir, later after his PhD, joined Purdue University for his postdoctoral research, and he has done his postdoctoral at Purdue for 2 years, and later he has continued his postdoctoral in California Institute of Technology, USA, and he has total 35 years of experience in neurobiology, genomics, and biochemistry of calcium signaling. He has won several awards and honors, like C.V.H. Rao Memorial Award for the best PhD thesis in Indian Institute of Science, Bangalore in 1992. He has also received several fellowships like visiting scientist at Hale
School of Life Sciences, Dr. Chavan and Dr. Zore and all the others for inviting me for this function and giving me this platform to talk about what we do in our institute. I have been to this university in the past, at least 2 times before, and has had the opportunity to visit the university and also be part of some conferences that were organized in the past. It was it is really nice to see that the university is growing as Honorable VC has elaborated. The university is growing in a major way. So I hope in this part of the country, this institution stays as a temple of learning, supporting all the aspiring students in and around. Now, getting to the theme of this conference, advances in biomolecular functions, advances in bioactive molecules. When we these days, when we talk about biomolecules or biomolecule, if you say biomolecule, it is any molecule which is present inside the body. But when you say bioactive molecule, although every molecule inside the body is bioactive, it is really having an activity inside the biological system. We are talking about molecules which you take from one species and put it into another species and then get some effect. So, for example, plant-derived compounds or compounds from microorganisms, marine organisms. So we find bioactivity in some other organisms which are supposed to be favorable or which are supposed to be therapeutically useful. So, this is a good activity, good scientific activity, because this permits us to find new medicines or new therapeutic approaches for many disease conditions. But at the same time, the various the opportunities and the promises that this field offers is not new to most of us. Bioactive molecule research has been going on for a long time. But I just want to point out one concern that I have felt over the years working in this area, which is one of the areas that we work, is that since we have all been thinking of getting an active molecule, then the immediate next thought for most of us is when it is going to become a drug. So we start talking about developing it into a drug. It has this potential, that potential, it will become a drug. But for a drug, for a molecule to become a drug, there is a long process. There are lots of bottlenecks or hurdles that it has to pass through, and it also involves huge amounts of money, and that is usually not within the reach of academic institutions. So their industries have to be involved, and industries have to be able to come forward to collaborate with any new biomolecules that are that have been discovered. So, which means that industries, to some extent, will have a say in the further progress of your research, bioactive molecule research. So, and government also has of late become very much dependent on the opinion of the industry in supporting bioactive molecule research. So, but for industry, the concern is most often profit and not necessarily human health, welfare, etc. They only if it is sustainable for them, they will support all the other causes. So therefore, it becomes primarily a profit-oriented endeavor, and anything that doesn't fall into that groove, any research activity, even if it is scientifically very sound or it has potential, if it is not appreciated or understood by the industry, it will have severe difficulty in that research in going forward. So this in other countries like US, I think there are government agencies which can take forward a bioactive molecule to the level of taking it up to the drug. For example, National Institute of Health they conduct clinical trials by themselves without the involvement of industries so far. So now government has announced doing clinical trials, but we have to wait and see how far and how long it will take for the new endeavors to become very effective. So, so that is one concern that I would like to express, that bioactive molecules becomes bioactive molecule research becomes the profit motive seems to become a major determinant in the progress of bioactive molecule research. So I, with this point to make, I would go into what we do in terms of bioactive molecule research. So today I thought I will speak about one approach in bioactive molecules, which has both promises as well as challenges, and that is multi-targeting, making a molecule target multiple biological pathways. So that is why I have given the title as towards achieving multitasking in drug therapy. So multitasking is basically at the same time, it's the same person doing multiple things. Similarly, having molecules developed which will go and act on multiple targets and then do multiple things inside the organism, inside the body. So that is basically the approach. And I will describe the research activity that we have been doing towards multi-targeting of molecules, of potential drug molecules, and then try to tell you how we have tried to do that. So my institute is in Thiruvananthapuram. It is a Department of Biotechnology Institute, Government of India DBT Institute. So I have been working in this institute for about 25 years now. So we have a neurobiology division. Our main area of activity is neuroscience, and one component of our activity is bioactive molecule research or drug discovery. Can we go to the next slide? Yeah, so this is what I have planned in the lecture today. As I said, our area of activity is neuroscience. So I'll just talk about neurological diseases in general, particularly Alzheimer's disease, some of the challenges that are faced in dealing with Alzheimer's disease, and then go into the idea of multi-target directed ligands or compounds which can be targeted to multiple drug targets. Then I go into a specific molecule called Tachrin and how that can be made to a multi-target directed ligand. And then I will show you our data, which started with modeling and then some in vitro assays, and then finally with an in vivo, with some in vivo experiments. Next, please. So can we reduce the slide? It's slightly missing on top and bottom. The title is missing, and then no, the screen view, it is going out of the screen, right? So is it possible to? While getting ready, I can proceed. So Alzheimer's disease is a neurodegenerative disease. I'm sure most of us know by now. It is been very much talked about. It is a major healthcare challenge in this disease, in this class of diseases, neurodegenerative diseases. The brain starts losing neuronal cells. Neuronal cells start dying. So there is gradual degeneration of the brain tissue. And as brain tissue degenerates, the functions supported by those areas in the brain are impaired. So in Alzheimer's disease, the areas that are severely impaired are what are the higher order brain function areas, or the cortex, frontal cortex. And frontal cortex is the area which supports memory, learning and memory in a major way. So one of the major symptoms of Alzheimer's disease is memory loss. People start forgetting their memories slowly, and the loss becomes gradually increasing to an extent that it starts impairing their normal functions, and they become totally unable to function by themselves. So in Alzheimer's disease, there are three major impairments. In addition to others, there are three major impairments that happens in the tissue, and they are impairments in the three pathways that are mentioned here. One is cholinergic signaling. Next is glutamatergic signaling, and the third one is amyloid protein processing. So these are three different signaling pathways that are happening in the brain, of which I'll be focusing on glutamatergic signaling in today's my presentation. So cholinergic and glutamatergic signaling involves can we move that out of the picture? Yeah. one of the major molecular players in excitotoxicity is Nmda receptor. Blocking Nmda receptor could be a major way by which excitotoxicity can be blocked. Next slide, please. Go ahead. So amyloid processing, we have already talked. Right. So, so it will be therefore advantageous to have blockers of Nmda receptor in preventing neurodegeneration. Now, so you can develop drugs, you can develop molecules which block Nmda receptor. Now, when you develop a drug, if we can develop a drug which blocks not only Nmda receptor but other targets also, for example, other targets which are relevant in this disease. In this case, we have seen amyloid processing as well as cholinergic signaling. There are targets which can be inhibited so that the disease can be reduced. And those two targets I have already mentioned, beta secretase and cholinergic signaling pathway acetylcholinesterase. So if you can develop a molecule which also has additional activities, which can go into these, which can target these other molecules, then that would be a more comprehensive treatment approach for this disease. So some of the advantages and disadvantages of such multi-targeted molecules are listed here. One of the major advantages is that drug-drug interactions encountered in combination therapies can be avoided. If supposing you develop single molecule, single targeted drugs, then in a disease condition like Alzheimer's disease, you have to give some 2-3 drugs. You have to give a drug for Nmda receptor blocking. You have to give a drug for cholinergic blocking. So these drugs which are being given, sometimes conditions where these drugs should not interact and cause toxic effects. So new molecules which are being developed should cause drug-drug interaction with the other molecule that are needed, other drug that are needed for that condition. Reduced off-target interactions. When you have a single molecule directed drug, sometimes it may have additional targets which we plan or which we did not design, what are called off-target effects, which very often would be harmful or which very often would be not favorable. So such off-target effects can be reduced if you use a multi-targeted drug, because you would be trying to combine more than one activity into one molecule, and you will be checking the activity of this molecule on very many targets. So thereby, there is a good chance that you will reduce off-target effects. However, the process of drug designing becomes more complex. That is a challenge. So it is not easy that you make a molecule which has different structural features that can bind to two different target molecules. So drug designing becomes complex, but the advances in bioinformatics and structural biology these days have made it possible to do this in a very pretty successful manner. And most importantly, the drug development process is simplified. Because as you know, when you have a bioactive molecule, then you have to show that it works in vivo. And then the major part of drug development is beyond that. So you have to show that the molecule has no toxicity at the animal level. Then finally at the human level, you have to show that the molecule still maintains its activity, significant amount of activity at when it reaches the human level. So for each drug development, this process takes about 10 to 15 years. So if you are developing two different drugs, and it involves huge amount of money. So millions or billions of dollars are required for developing one drug before it becomes clinically usable. So if you are developing two drugs for two targets, the total amount of time, expenditure, maybe you can parallelly do it, but the amount of expenditure required would be doubled. But if you can design a molecule with more than one activity, say 2 activities in one molecule, then this step, safety profile, and efficacy profile assessment in higher systems like animals and in humans can be reduced. And it becomes half, or it becomes reduced by according to the number of activities you are putting into a molecule. So that would be a major advantage if it is possible to have a successful multi-targeted compounds. So the compound that we chose for our study, or we started with, is a molecule called Tachrin. The title is there on the top, Tachrin. So this is the first drug approved for Alzheimer's disease. So the first drug approved against Alzheimer's disease but it did not stay as a drug because it had some severe side effects. So this molecule was developed as an inhibitor of the first pathway, cholinergic pathway. It blocks acetylcholinesterase, an enzyme which degrades acetylcholine and thereby reduces amount of acetylcholine in the brain. So if you block that enzyme, then degradation of the neurotransmitter acetylcholine would be reduced. So acetylcholine level will go up, and that is something that is needed in Alzheimer's disease. So cholinergic blocking is one of the strategies used in Alzheimer's disease. So this was a cholinergic pathway blocker or acetylcholinesterase blocker. It was also a weak antagonist, the other molecule that I described a little bit in detail, the glutamatergic signaling molecule. So the Tachrin molecule has weak antagonism against Nmda receptor. Now, although this molecule reached the clinic, it was found that it had some side effect which was not detected during the drug development process. So it had hepatotoxicity. So it was withdrawn from the market because it had withdrawn from the clinics because it had hepatotoxicity. It had toxicity on the liver. So we thought, why not take this molecule and then start doing some modifications in this molecule and then see whether we can overcome this toxicity, as well as can we improve its activity against Nmda receptor. So the rationale for thinking of that approach is that if you reduce the toxicity, one thing that you can do is you can reduce the dose. Any molecule would be toxic at higher dose. Even a biological molecule, even a molecule which inside our body, if it is given in excess, it can be toxic. So the so-called normally used drugs, non-toxic drugs, are also non-toxic at a particular level of dose. A dose in which their activity is optimal. So if you can increase for a molecule which is showing toxicity, if you can improve its activity, then you can reduce its dose. And if you can reduce its dose, can we reduce the toxicity? So that was the approach. So can we reduce the toxicity by reducing the dose? And how to reduce the dose? Can we increase the efficacy of this drug, its activity? And also, can we engineer this additional activity? Antagonism towards NMDR was very weak in that molecule. Can we enhance that? So that was our approach. So what we attempted is to improve its target interaction. So this molecule's interaction with acetylcholinesterase and Nmda receptor, can we improve its interaction? But at the same time, maintain its normal pharmacological properties, ADME properties. Since it is already a molecule which has reached which has gone through drug development, we know that at least this basic nucleus has favorable pharmacological properties or ADME properties: absorption, distribution, metabolism, and excretion. So any drug molecule should be very well absorbed. It should get distributed very efficiently in the body, and it should be properly metabolized, and it should also be excreted from the body efficiently. So these are essential requirements for any molecule. So this molecule has those properties. Right. However, the hepatotoxicity that led to its withdrawal. So since we know this molecule has good ADME properties, derivatives of this molecule may also have good ADME properties. So there is a good chance that they will not be having any poor ADME properties. So that is another reason why we chose a molecule which was already tested in the drug development process. Next slide, please. Right. So, our main aim was among the possibilities of enhancing its activity. Our main approach was to enhance its activity against Nmda receptor, the glutamatergic receptor that I talked about. And this receptor is a multimeric receptor with multiple subunits, which are shown here. All these are glutamate receptors, of which what is shown here is the set of subunits which will make Nmda receptor. It's a tetrameric receptor. Different combinations of these subunits will make the receptor. Next slide. Nmda receptor antagonists are already known. People have been trying to develop Nmda receptor antagonists, but there are some major issues. So far, the many inhibitors that were developed, they had severe side effects because they interfere with normal functions, which I already mentioned, because NMDA receptor is also necessary for learning and memory. So it should not, blocking that should not affect normal functions. And efficacy of developed NMDA receptor inhibitors have been not great. They have been, efficacy has been modest. And the number of drug molecules available or the number of inhibitors that were generated against NMDA receptor has also been less because doing NMDA receptor assays is difficult. Any inhibitor development would very much depend on the activity assay for the target molecule. You are developing an inhibitor towards a particular enzyme, you should have an assay, enzyme assay, and then you check your target molecule, drug molecules in that assay and see whether that molecule is inhibited. So if the assay is very difficult, then checking several molecules on the assay becomes time consuming. It can become very expensive. And NMDA receptor is a molecule for which assay is a difficult assay. And therefore, the number of molecules that could be screened with an NMDA receptor assay has also been limited. And therefore, the number of drugs available has been limited. So because of all these reasons, we don't have many good NMDA receptor directed drugs. So in our lab, from other activities that were going on in the lab, we were able to address this issue, that is the assay for NMDA receptor. We were able to develop a new assay for NMDA receptor, which is a simple and easy assay. Next slide. Go to the next slide. Go ahead. So this go ahead. Next. Yeah, this is to show that if you start with several inhibitor molecules in your primary screening of an inhibitor, then as the process of drug development proceeds, the number of many of these get dropped out because of various side effects, and finally you may reach with one successful molecule, for which you have to start with a large number of primary hits. So generating large number of primary hits is essential, and that again depends on the assay method that you are using. If the assay method is difficult, generating large number of hits becomes difficult. Next. So for NMDA, go to the next. So yeah, okay. So for NMDA receptor, until recently, NMDA receptor structure has not been available. Crystal structure or three-dimensional structure has not been available. For modeling-based drug discovery, you need the structure of the target molecule. So if the structure is not available, then that approach is not very much possible. So therefore, the other approach is high throughput screening of compound libraries. That is something that can be used for any molecule for which, even if you don't have the structure. So for NMDA receptor drug discovery, this has been the major approach until recently. So only about 2-3 years back, the structure of this NMDA receptor became available. Next slide. So the existing assays until we developed our assay were electrophysiological measurement or patch clamp measurement, with which you measure the calcium current which is allowed by this receptor, or by real-time calcium imaging. You image calcium levels inside the cell by using fluorescent dyes, and then use that as a measure of calcium channel activity. Both these assays are what are called real-time assays. These assays have to be measuring the activity as and when the enzyme is working in the live cell. So you have to maintain the cells in live condition, and then you have to activate the channel, and then have an equipment or an assay method by which the increased calcium levels in the cell or calcium current through the channel will have to be measured. So these are what are called real-time assays. Real-time assays have some advantages, such as they are very powerful. They have very high temporal resolution. They are very fast, and they have very high spatial resolution. They can resolve subcellular regions. Small changes happening in different regions of a cell can be detected. However, they are usually very expensive, because being very powerful assays, the equipment required will also be very powerful. Obviously, the equipment is going to be expensive. So the assays that I mentioned, the real-time assays, are therefore expensive as well as difficult to scale to high throughput. That means screening large number of molecules is also difficult. Next. So we developed an assay, which is a non-real-time assay, which is not a real-time assay, in which you can do the calcium channel activation, the calcium signal induced, calcium signal induced memory is formed in the cell, and that can be checked at a later stage. You don't have to measure it when the cell is live and active. So it's what is called as an endpoint assay. So it is an assay in which an endpoint is. Can you excuse me? Can you push this up? No, not next slide. Go back to the previous slide. But that panel, if you can remove this panel. If you can move that panel. Yeah. So what we developed is an endpoint assay for calcium sensing. And what we depended on is a protein-protein interaction that happens inside the cell in response to calcium. So next slide. And the principle of our assay is shown in the next slide. Next slide, please. When Nmda receptor is activated in the synapse, the same slide as the same scheme that I shown before, but in a different way. When Nmda receptor is activated, calcium comes in, it activates a protein called calcium calmodulin activated protein kinase 2, CAMK2. Upon activation, this protein will act on its substrates, but it will also translocate to Nmda receptor and bind to a subunit of Nmda receptor in a very stable manner. And when the calcium signal disappears, normally, if you put a calcium dye, the fluorescence from the calcium dye also will disappear. However, this interaction, being a very stable interaction, it would persist. So calcium signal would induce the formation of this complex, but the disappearance of the calcium signal would not affect this, that would stay. So in other words, this complex would be a memory of a previous calcium signal. So if we see that this complex is formed, that means you can conclude that there was a prior calcium signal. So since this is stable, you don't need to do real-time measurements. You don't need to measure it in the live condition. You can do the assay and then take it and then measure and look for the presence of this complex at a later time. So therefore, this is an endpoint assay. It can be done very conveniently with very simple methods. You can use just a normal fluorescent microscope and do the assay. Next slide, please. So what we have done is we have put these two proteins in a heterologous cell system. What I have shown was a neuronal system. We put both these proteins into HEK293 cells, human embryonic kidney cells, which do not have endogenous expression of these proteins. So CAM kinase is labeled with a GFP, green fluorescent protein, and its binding protein, Nmda receptor subunit, the binding sequence alone is added with a mitochondrial targeting sequence, so that when it is expressed in the cell, it will go and bind to the mitochondria. And to this system, but CAM kinase would be in the cytosol. If you give a calcium signal, CAM kinase would be activated and it will now move to the mitochondria because its binding partner is there around the mitochondria. You'll go and bind to the mitochondria and then aggregate around the mitochondria. In the next slide, go to the next slide, please. No, previous. Yeah. So what we will see is that in the cell without activation, you see the GFP tagged protein CAMK2 distributed in the cytosol. Upon activation, when it is activated, it will move towards mitochondria and then form aggregates around mitochondria because of its binding with the other protein. So you will get this kind of an aggregate pattern, a punctate pattern because of formation of these aggregates. So maybe we can switch off these slides. This set of lights. The punctate appearance that is formed in these cells is an indication of calcium influx into the cell. And here, what we have done is we have treated the cells with activators of Nmda receptor, which is glutamate, glycine, and calcium. Calcium has to go through glutamate and glycine are activators of the channel. Yeah, you can switch off these slides. Right. So I hope it is better now. Visibility is better. So, so you can see this punctate appearance, and that is very different from these non-activated cells. Here we are treating the cells without calcium. So without, if there is no calcium to go in, the CAMK2 will not be activated. So you will not get this punctate pattern. Now, if you do this activation in the presence of inhibitors of Nmda receptor, there are already known standard inhibitors of Nmda receptor, such as AP5 and MK801. These are known inhibitors. So the so we sort of found a solution to one of the problems in the drug discovery of Nmda receptor, which is having an easy assay for doing Nmda drug screening. So having that assay with us, then we went for a little bit of drug designing. And now this is done by my collaborating group. I have two collaborating groups who did the bioinformatics part of it, the drug designing part of it. One is from Kannur University back in Kerala, and the other group is in Japan, in Riken. So both these groups contributed to us designing drugs. So the major steps in their designing is listed here. So they docked the Tachrin structure to Nmda receptor structures, multiple structures of Nmda receptors. There are structures are also not stable. When you simulate this, changes in the structure would, movements in the peptides would give rise to different structures. So they did what is called as ensemble docking of Tachrin to Nmda receptor, and then identified some structures of Nmda receptor as suitable candidate for further drug development. And they also calculated binding energies. Then using the complex, they found that there are additional points of contacts which can be developed between Tachrin and the molecule using the docked structure. And using the docked structure, they designed about 75 derivatives of Tachrin, which can have additional interactions with their targets. And then these derivatives were again subjected to ensemble docking with the structures, and all the 75 derivatives are shown here with their binding energies in the heat map. So they calculated the heat map and the binding energies for all these molecules, and their ADME properties were also predicted by in silico tools. There are tools by which ADME properties can be predicted. And then based on all these considerations, among these 75, we selected about less than 20 molecules for actual testing, because it is not testing the 75 is still a difficult task. It is still possible with the easier assay that we have, but there were very obvious reasons by which many of them can be ruled out. So thereby we finally ended up choosing about less than 20 molecules. And then they were tested in the assays, biological assays, in vitro and in cell culture assays. So the derivatives that were designed fell under three groups. Group 1, which were derivatives of this part, R1. Group 2 were derivatized in this part alone, and group 3 were derivatized in both the points. And derivatization was done with many different substitute molecules. Next slide. Yeah, and here is the data of in vitro screening of these derivatives. They were first screened against acetylcholinesterase, the original target against which this molecule was developed, Tachrin. Acetylcholinesterase, the cholinergic signaling which I developed initially. And we found that this is Tachrin, the original molecule. Its inhibition is taken as 100%. I believe it is 100. And compared to that, all the derivatives were normalized, their inhibition. And you can see that most of them maintained their inhibitory activity. Some of them were a little better, and some of them were, so they suffered a little bit in their inhibitory activity. So by designing, the original inhibitory activity should be maintained. So that was one of our aims. So by and large, we were able to maintain. There is some fluctuation here and there, but by and large, it was maintained. Their activity on another target of the cholinergic pathway, butyryl cholinesterase. This is not a major player of the pathway, but it's still a significant player. So the blocking butyryl cholinesterase also has been shown to have therapeutic effects. So our derivatives were having effect on the butyryl cholinesterase also. Their original activity is present, but not to the same extent. Then they were tested on Nmda receptor activity, which is the assay that we developed. And we found that here is the original compound Tachrin. This is basically percent inhibition, and Tachrin has very minimal inhibition on Nmda receptor. But many of the derivatives were very highly inhibitory. So the designing has really worked. So the way it was designed by docking to Nmda receptor, many of the derivatives are showing actual inhibition of Nmda receptor. And they have been much better than the original molecule Tachrin. And their IC50s, among the molecules which are inhibitory, their IC50s were calculated again using the same assay. And IC50s shows that some of the molecules were very, very potent, about 400 to 500 fold more potent than the original molecule Tachrin. So we have a set of molecules which, in addition to their cholinergic inhibition, they are also inhibitory on Nmda receptor very effectively. So we have a set of multi-target directed ligands, or at least they have action on 2 targets. Now, one of the major complaint against Tachrin was that it was hepatotoxic. It was toxic to the liver. So there is an in vitro assay for hepatotoxicity, which is basically a liver hepatocyte derived cell line called Hg2. People use this as an in vitro hepatotoxicity assay. And we tested all these compounds on Hg2 cells, whether they are toxic to Hg2 cells. And we found that most of them, not most, all of them are not toxic or less toxic than Tachrin. Tachrin has more toxicity compared to all of them. They were, for the concentrations that we tested, I think we tested 4 concentrations. For each, most of the derivatives were not toxic, even at the highest concentrations that we tested. So the derivatives, therefore, did not increase the hepatotoxicity. That is also possible. When you change the molecule, hepatotoxicity which was observed can also increase, but that did not happen. So that was also favorable. So that was not by design, that just happened by default. So, all right, can you tell me how much time I have? 5 minutes more, okay. All right, so, so we have now a set of compounds acting on acetylcholinesterase and Nmda receptor, at the same time not having any major increase in their hepatotoxicity. Next slide. So we then tested them on neuronal cells. So far we have been testing them on non-neuronal cells. Now we tested them on neuronal cells. If you treat neuronal cells with glutamic acid, which is an activator of Nmda receptor, it is known that because of excessive activation of Nmda receptor, neuronal cells will die because of excitotoxicity. And you can see that neuronal cells, when treated with Nmda receptor, are undergoing death. There is significant death in neuronal cells. But if you block Nmda receptor along with glutamate treatment, this neuronal cell death can be prevented. So here, what is used is a standard inhibitor called MK801. So any inhibitor of Nmda receptor can block this. So we now tested the compounds that we developed, and they are all able to protect neurons compared to their absence. So in the absence of, if you treat with glutamate alone, neuronal cells die, but in presence of the compounds, the cells are protected. So as expected, Nmda receptor blockage is protecting neurons from hyperactivation of Nmda receptor and the associated excitotoxicity. And we have, this is one assay that we use. We have used different, this is a imaging assay. Here is a quantitation for that. This is how glutamate induced toxicity causes reduction in cell number. These compounds are able to reduce the toxicity. We also did a couple of biochemical assays to quantify this cell death, MTT assay and then G6PD assay, glutamate, glucose 6-phosphate dehydrogenase assay. And in those assays also, we found that the glutamate-induced cell death can be prevented by these derivatives. So all the different assays show that in the neuronal cell, these molecules can be protective against neurodegeneration. Next slide. Next slide, please. Yeah, so we went to an in vivo model. There is a rat model in which you can inject monosodium glutamate or MSG. It's a food additive, food preservative, can also be toxic at high dose. As I said earlier, anything can be toxic at high dose. So MSG given at a little high dose can be damaging to the brain. That is already known. So we used MSG treatment to rats, intraperitoneal injection for 15 days. And at the end of this, towards the end of this, we started looking at the animal's behavior. If brain is damaged, brain functions would be affected. So behavior of animals tested in certain behavioral assays can be used to look for these brain damages. So we used a test called the Morris water maze test. Next slide, please. And in this test, what you do is there is a water tank in which you drop the rat, and the water tank also has a platform which is a little below the surface of the water. So the rats which are in the water would be uncomfortable and they would swim around and then they will try to come out, but they cannot come out because the walls are high. So after a lot of trial and error, they would finally find the platform and then they will be able to stand on it. The platform will not be visible to them because the water will be made opaque. So they have to find the platform by using other cues in the room, other signals in the room. Using that, they should be able to, they will finally learn which is the location of the platform. So this happens after repeated trials by putting the rat into the water several times. So for over 5 days. So each day we put them for 5 times, and by day 3, day 4, they would learn where is the platform. So as soon as you drop them, they will straight away go to the platform. And by day 5, you can see the same animal on day 1 was roaming around without having any clue where the platform is. But on day 5, it knows damn sure where the platform is. So it goes straight to the platform. That shows learning. So the time taken by the animal to reach the platform is taken as an index of its learning and memory. The time taken called escape latency. Now the monosodium glutamate treated animal, on day 1, both are same because both are starting to learn. But on day 5, their learning is still poor. Compared to the normal animal, the monosodium glutamate treated animal still has a little bit of doubt, or is still not learned properly. So that shows learning impairment, cognitive impairment, which is indicative of brain damage because of MSG. So we have other data showing that there is brain damage, etc., which I'm not showing. And then next slide. If you give an Nmda receptor inhibitor, can this be prevented? So we used a standard inhibitor called dextromethorphan, which is known to block Nmda receptor. So if you give dextromethorphan along with monosodium glutamate, you can see that the animals are fine. The monosodium glutamate induced impairment that you saw can be prevented if Nmda receptor is blocked. So Nmda receptor blocker can be assessed by using this assay. Now, we tested the compounds that we have, some of the compounds in this assay. The next slide. Go to the next slide, please. Yeah, this is just to show that the model that we used shows some molecular changes. For example, when you treat with monosodium glutamate, this protein called TrkB, which is a cell survival related molecule, is reducing. But if you give dextromethorphan, then monosodium glutamate is unable to reduce that cell survival molecule. So the molecular changes induced by monosodium glutamate and the associated brain damage are prevented if you give an inhibitor of Nmda receptor. So in the next slide, go ahead. Next slide. So there are other molecular analysis that were done, and we found that monosodium glutamate induced damage can be prevented if you also give an inhibitor of Nmda receptor. Now, so this assay therefore reasonably well assesses Nmda receptor in vivo, the Nmda receptor blocker function in vivo. So now when these compounds were given, you can see that the escape latency, the time taken to reach the platform, was increasing for monosodium glutamate, which you have already seen. Monosodium glutamate treated animals took a long time. But along with that, if you give the compounds, the monosodium glutamate treatment is not increasing the time taken. So it is preventing the animals impairment, cognitive impairment. So they were tested at a higher dose of monosodium glutamate treatment, and you get the same result. You can see that saline, this is the normal animal learning by day 5. Monosodium glutamate treated animal still not learning on day 5. Then monosodium glutamate and the compound, one of the compounds, compared to here, there is improvement. And that is what you see here in all these graphs. There is improvement in learning in the animals when they are fed with the compound along with monosodium glutamate treatment. So we are at this stage now. We have developed a set of multi-targeted compounds. We have tested their multiple activities in vitro and their glutamatergic activity, their Nmda receptor inhibitory activity, which we developed newly in the compound, that was tested in the in vivo condition. So we need to test whether their other activities, such as cholinergic activity, which was its original activity, whether that is being maintained. That needs to be tested. But at least in this animal model, they are definitely helping in the improvement of the animal's brain damaging condition. Next slide. So to summarize, multi-target directed ligands is what I tried to introduce in this bioactive molecule conference. Multi-target directed ligands are an approach towards molecules, developing bioactive molecules with advantages and disadvantages. So they have several advantages, and Tachrin was used to derive several multi-target directed ligands with significant efficacy, and they were shown both in vitro and in vivo. Now you can skip a few slides. Go ahead. Now we are, I'm sure the 5 minutes also has exceeded. So yeah, go to the next slide. So I would like to acknowledge my colleagues. These are my students over the past several years who have contributed to this work. He is my scientist colleague in my institute who have helped me with animal experiments. These two are the bioinformatics groups who did a very excellent job of designing these molecules. Kannur University and then Riken, Japan. And then this group in Guntur, Andhra Pradesh, VFSTR. Dr. Sheikh Anwar and his student Koti Reddy, they actually synthesized the molecule. They were our chemists. And then this is technical support in my own institute, doing all the imaging. And here is funding. And thank you all for your patience. So there are questions. Yeah, I will be happy to take questions if time permits. Okay, sure.
Now the topic is open for discussion. Any questions from students?
Yeah. Yeah. The patient-to-patient variation is not being addressed here. The major aspect, which is the disease mechanism, has different target, multiple impaired pathways. So if in the disease, its effect on different patients might be varying because of variation in its genotype in the sequence of NMDA receptor. Minor variations between populations might be causing difference in the effects. So those variations should be taken right at the in vitro level. So if we start developing drugs like that, maybe we will also have the information of how this drug would perform on different populations. So that's a very important point, which I think is, I don't know whether it is being practically being implemented at this stage, but it is definitely necessary, and it will become an essential thing very soon. Yeah. Oh, yeah. Yeah. Yes. Mm-hmm. Mm-hmm. Yeah, can we go to that slide? In that slide, I have also listed which were more effective. Yeah. Mm. Okay. Yeah, see, that's a point that I did not mention. All the Ayurvedic formulations or the traditional medicines are not pure compounds. They have multiple, those are formulations of multiple compounds. Maybe they are doing this multi-targeting. Any disease would require multi-targeting. So you need to, no disease is just happening because of one pathway being affected. Mm-hmm. Mm-hmm. Separately, separately, and you are saying to give those compounds individually. Yeah, but but the point is, if you have two compounds, then drug discovery becomes twofold more difficult. The regulations and drug of no. The regulations of drug discovery in getting a drug approved, we have two compounds, it is double the expense. $1 billion becomes $2 billion, or $5 billion becomes $10 billion. So that becomes a major issue. So that is one of the major advantages of multi-target directed ligand. So yeah. Yes, yes. Yeah, in my institute there are two. Since I'm thinking about the master's students, masters and bachelor students. For masters and bachelor students, one thing is your internship. So our institute supports internship, but that is with a fees. That is a paid course, 3 months, 6 months, whatever duration you want. You can choose some lab, you can look at the lab and its activities, and then depending on your interest, you can apply. And we have some regulations. At a time, one lab can support only 5 students or 6 students. So if a lab has vacancy, then you, and if the time that you need, your requirement matches with the lab vacancy, you can get into the lab. You have to send the CV, and the lab supervisor will look at your biodata, and then if there is proper match, it's not very difficult internship. But then for master's student, then you also have PhD. The PhD admission is the basic qualification is national level fellowship, CSCR, UGC, DBT, ICMR. You have to have some fellowship, and only those with fellowships can apply, and they will be screened again. There will be an interview, and some people will be selected from there. So that is how the PhD selection, but that is a national level selection. Both these are national level selections. So the website is available. All the details are there on the website. So internship could be a good idea which can accommodate more people. So you get an exposure to the whatever activity is going on. We have a lot of, being a DBT National Institute, we have a lot of equipments, a lot of facilities available. So students would get, it's a good exposure for students. Then when it is PhD, then it is a little more difficult. You need to compete.
Thank you very much, sir. Thank you for sharing your great knowledge in achieving multitasking drug delivery. And it will be a great pleasure for us to listen you again in our future events, sir. Thank you very much. Now, may I request Professor T. sir to felicitate Dr. R.V. Om Kumar, sir. Sir, please. Thank you very much, sir. Thank you. Now, it's high tea, and I would like to request all of you to move outside of the Senate Hall. Tea is waiting for you, and we will resume here after 10 minutes. You can enjoy high tea. We will resume here after sharp 10 minutes because already we are getting late. So again, there are 2 lectures in this session, and after that lunch break will be there. So you have to resume it in 10 minutes.